Hi, my name is Greg, and I'm a fifth year in the Zhuang lab. My research focuses on the area of ubiquination involved with the DNA damage response pathways. I am interested in the cellular response to DNA replication fork stalling as a result of DNA lesions. PCNA, shown in the slide, becomes ubiquinated or polyubiquinated in response to the stalling. One attachment of ubiquitin to PCNA leads to translesion synthesis, and a polyubiquitin attachment to PCNA leads to a less understood error-free pathway. In the Zhuang lab, we have developed a chemical ubiquitin ligation strategy that allows us to probe the inner workings of this pathway. We have utilized our microacceptor containing linkage to determine the preferred ubiquitin site of PCNA by the deubiquinating enzyme for the yeast PCNA, UBP10. We have also utilized multiple chemical ligation strategies of ubiquitin in combination to uncover the preferred linkage types as well as endo versus exo deubiquinase activity. Our future research in the Zhuang lab is focused on utilizing our ability to generate polyubiquinated PCNA for investigatory studies to uncover mechanisms and proteins involved with the error-free DNA damage response pathway.
As a third-year graduate student in Dr. Zhuang's lab, my project focuses on the role of ubiquitin in mitophagy, the selective degradation of mitochondria. An overwhelming number of studies have linked mitophagy to neurodegenerative disease states, notoriously Parkinson's disease. When mitochondria undergo a depolarizing event, outer mitochondrial membrane proteins become ubiquitinated, signaling for mitophagy to occur in an attempt to rescue cell viability. Two deubiquitinases are implicated in removing these ubiquitin chains from damaged mitochondria, indirectly opposing Parkin, an infamous E3 ligase. My goal is to develop an assay to allow for the quantification of these DUBs inhibition for inhibitor discovery. This project utilizes a similar cell-permeable probe generated by Dr. Weijun Ji, combined with the lysate-based alpha-LISA assay developed by Dr. Christine Ott, both previous members of the Zhuang lab. To summarize, we intend to identify a selective DUB inhibitor to function as a possible therapeutic in Parkinson's disease.
My name is Christine and I'm a fifth year in the Zhuang lab. This project focuses on studying ISG15 and immune response. ISG15 is a ubiquitin-like protein. It can function as a post-translational modifier, but it has also been shown to act as an extracellular signaling molecule. In the schematic, you can see that upon viral infection, ISG15 can be conjugated to viral proteins through a 3-enzyme cascade, mirroring the ubiquitination process. Interestingly, ISG-elation of viral proteins has been shown to inhibit viral replication, making this a key facet of antiviral response. ISG15 has been shown to interact with viral proteins non-covalently as well, to sequester viral proteins and thus inhibit viral replication. In the Zhuang lab, we're studying these different interactions through novel ISG15 probes. Activity-based probes will allow us to identify proteins that selectively reverse ISG-elation by removing ISG15 from substrate proteins, while interaction-based probes allow us to elucidate protein partners that are not targets of ISG-elation, but rather interact with free ISG15. SARS-CoV-2 PL-Pro is the d-ISG-lase in COVID-19. It functions to antagonize the ISG-elation process and promote viral replication. With our probes, we are able to study the interactions and activity of this enzyme and use that information to aid in the development of antiviral therapies, such as small molecule inhibitors of this viral enzyme. Through chemical biology and biochemistry techniques, this project aims to elucidate the different pathways and interactions that ISG15 is involved in.
As the newest member of the Zhuang lab, my project will be focusing on a newer class of drug therapeutics known as PROTACs, proteolytic targeting chimeras. These molecules work to preferentially tag proteins of interest for recruitment to the proteasome for degradation. They are designed to recruit both a protein which will be ubiquinated for recognition by the proteasome, as well as the corresponding E3 ligase required for ubiquitin tagging of a protein of interest. These two protein-specific moieties are joined through a linker region that serves to bring the proteins within close proximity for this modification to occur. Restraints in this field of research existed because few small molecule ligands had been developed to bind ubiquitin ligases, and their small peptide counterparts were not ideal as cell permeability is extremely difficult due to their size. The development of small molecule ligands that bind the E3 moiety with high affinity brought with it increased potency of these molecules, paving the way for PROTACs as a new class of drug therapeutics.